Hi, this is Peter Glenn Chua, and this is my lecture on cell wall disruptors and translation inhibitors, the first of two lectures on antibacterial agents. This lecture is prepared for the Ateneo de Manila University, the School of Medicine and Public Health. For this lecture, we will be tackling several drug classes that are broadly categorized based on their mechanism of action. The first is for cell wall disruptors. These are drugs that cause the bacterial cell to burst by weakening the cell wall and/or bacterial capsule. The second category is for translation inhibitors. These drugs slow down the metabolism of bacteria, allowing the immune system to be more effective in capturing and destroying the bacterial cells. As an introduction to some of the most important drug classes that you will use as a physician, besides the historic and civilization-defining achievements of these drug classes, we will not be focusing on dosage and preparations, but look more on the why and how these drugs are able to control bacterial infection and enhance the effectiveness of the immune system. For this first lecture on antimicrobials, we will be covering beta-lactams, chloramphenicol, glycopeptides, streptogramins, tetracyclines, macrolides, oxazolidinones, and lipopeptides. Other drug classes, including aminoglycosides, sulfonamides, and quinolones, will be discussed in the lecture to be given by Dr. June Cuenca. Notice that for this particular slide, there was a burst of discovery around the 1940s to the 1960s. This was inspired by the series of wars which started from the First World War and spilling over to the Korean and Vietnam Wars. And wars have been known to trigger major advances in medicine and surgery, including the discovery of antibiotics. The fact that the last 50 years have been among the most peaceful in human civilization, we see that less and less antibiotics are being discovered and developed. And this is one of the problems that we need to address as we encounter the problem of antimicrobial resistance. And so we jump to the antibacterial drug targets of known drugs. It is important to understand that the prokaryotic cell wall is the target of a vast majority of antimicrobial classes. The cell wall is a major differentiation between animal and bacterial cells. It is very stiff in the sense that it is able to resist high internal osmotic pressure. Inside the bacterial cell, there is a high concentration of solutes. There is plenty of nutrients, plenty of sugar, plenty of salt, and so it continues to pull in water. And this oncotic pressure lends to an increase in osmotic pressure. And so this highly pressurized internal environment is one of the forces that drive the fission of bacterial cells. A properly working cell wall is able to control and harness the osmotic pressure so that it is used to propagate cells, so that it is used to push for binary fission, to push for meiosis. A proper bacterial cell wall is able to determine the shape of the bacterial cell. Depending on the contents of the bacterial cell wall, we can see bacilli, we can see cocci, we can see spirochetes. And depending on the shape of the cell wall, the weak points of such a cell wall are also the points wherein the bacterial cell is most likely to burst open. The bacterial cell wall, in relation to inflammation and the response of the human body, contains virulence factors. These virulence factors enhance the ability of bacteria to cling to living tissue and therefore to cause disease. These will include the ability of certain bacteria to form biofilms. Uh, Bacteria as a cell um, is not very virulent by itself, but as a community or as a bacterial colony, certain virulence factors are expressed. And it is only in such an environment, in a colony or in a biofilm, an ecosystem, the biofilm is actually a form of bacterial ecosystem that fosters the maturity of bacterial cells and therefore allow the expression of certain virulence factors. One important component of the bacterial cell wall, of the prokaryotic cell wall, is peptidoglycan or murine, which is more prevalent or more predominant in gram positive than in gram negative bacteria. One molecule that is notable for mycobacteria, that is the genus of your bacteria that causes tuberculosis, are the long mycolic acid chains. If you can imagine a plush carpet, those furry carpets that have been popular for the last 5 years, um, you can imagine your tuberculosis bacilli to be wrapped in such a fur coat, in such a shaggy carpet. Because these long mycolic acid chains are very, very long. And they would render the appearance of the tubercle bacilli as, like I don't know, a yeti or perhaps a woolly mammoth. In such, it's like a hairy tubercle bacilli. And as such, um, it makes mitosis and meiosis quite tricky for mycobacteria. That is why mycobacteria are notoriously difficult to culture, because the cell wall is very shaggy, therefore quite heavy and quite stiff to break open and allow the cell to split. And understanding that cell wall is important to controlling bacterial growth, to killing bacteria. And that is an important concept that we will encounter throughout this lecture. To further illustrate the importance of the cell wall or the uniqueness of the bacterial cell wall, we see here um, diagrams how the cell wall of certain microbes are assembled. The gram-negative bacteria have an outer membrane on which lipopolysaccharides are attached. Lipopolysaccharides are an important virulence factors. They themselves can be toxins, meaning the human body will respond to it quite strongly, and the response can be quite injurious, not only to the cell but to the host, to the person itself. And note that there is peptidoglycan in the gram-negative bacterial cell wall, but not as much as the peptidoglycan present in a gram-positive bacteria. In the cell wall of the gram-positive bacteria, you have a thick layer of peptidoglycan anchored on the cell membrane. The anchor can be a lipoprotein or it can be a lipoteichoic acid. Now, a recent study has identified the lipoteichoic acid to be an important molecule in the effect of antimicrobials. Lipoteichoic acids are the synthesis of lipoteichoic acids are inhibited when bacteria are exposed to penicillin or when there is a prolonged stationary phase as caused by protein synthesis inhibitors. And because the lipoteichoic acids are inhibited, there is an increase of wall attached teichoic acids. Now, these wall-attached teichoic acids promote the attachment of a molecule named LYTA, light A, which is an autolysin, meaning it actively breaks down the cell wall. And so, in producing more teichoic acid, wall-anchored teichoic acids, more autolysins are recruited to bind to the capsule and therefore facilitate the breakdown of the cell wall. This has been discovered only in the last in the last 2 to 3 years, and the identification of the LYTA promises to be a novel target for new antibiotics to target, and this is an important lead for us, for the medical community, as we look for more antibiotics to battle growing microbial resistance. As I've previously described, you can see that the mycobacterial cell wall is kind of furry or fuzzy with long strands of mycolic acids and with long strands of polysaccharides like lipoarabinomannan. Note that these shaggy strands of mycolic acids um, allow the mycobacteria to adhere to surfaces, but at the same time, they make it very tricky for the mycobacteria to actively split open. That puts the metabolism of the mycobacteria by default at a lower level because they are not able to absorb as much nutrients from the environment given that they have a very thick fur coat for a cell wall, and that the cell wall is kind of heavy to push apart. For comparison, we can see here the cell wall of fungi, which is composed of chitin, polysaccharide, uh, uh, glycopeptide, composed of beta glucans and proteins. Um, and for certain antifungal agents, chitin is the target. Inhibition of chitin synthesis also renders the fungal cell wall weak and therefore also destroys the fungal filament. So it is important for us to identify the molecular targets of certain antibiotics. Note that for beta-lactam compounds, which will comprise a vast majority of cell wall inhibitors, beta-lactam compounds include your penicillins, cephalosporins, monobactams, carbapenems, and beta-lactamase inhibitors. They target the penicillin binding protein, which is a transpeptidase. These transpeptidase allow the attachment of polypeptides to the polysaccharide backbones, which is the building block of your peptidoglycan. By inhibiting the penicillin binding protein, um, you are not allowing peptidoglycan to properly form the cross links, to properly lengthen, and therefore you are weakening the peptidoglycan cell wall, leading to a similar effect would be um, the effect of glycopeptides, which would include vancomycin and teicoplanin. Um, in approximating the shape, the appearance of the D-ala terminus, the alanine terminus of the peptidoglycan side chain, um, elongation of the peptidoglycan filament or the peptidoglycan polymer is inhibited by glycopeptides. Specifically, the vancomycin binds to the target with H bonds. Vancomycin is quite a large molecule. And so in wrapping itself around the D-ala terminus, it essentially wraps it around a glove so that no further elongation, no further attachment of polypeptides can happen. Therefore, it is inhibiting the elongation of the peptidoglycan backbone. One drug that targets the enzyme that allows the shuttle of the building blocks from the inside of the cell to the cell wall space would be bacitracin. It inhibits bactoprenol. Bactoprenol allows the shuttle of of peptides from the cytoplasm to the cell wall space, which is outside the cell membrane. And in inhibiting the bactoprenol, we are depriving the building blocks from reaching the construction site. Another target that essentially leads to disruption of the cell wall would be the enzymes alanine racemase and the alanyl alanine ligase. We have already identified the D-ala terminus to be an important part in the synthesis of the peptidoglycan, as demonstrated by the effect of vancomycin. And in changing the appearance of the D-ala terminus, we are also preventing the elongation of the peptidoglycan backbone and also the side chain. So the alanine racemase essentially prevents the conversion of L, the leftward oriented alanine, to a D-alanine. So that's one enzyme, the racemase effect. The ligase effect prevents the attachment of D-alanine to another D-alanine. Therefore, the D-ala terminus is not synthesized. And finally, we have enol pyruvate transferase, which is happening more in the cytoplasm more than in the cell wall space. What phosphomycin does is it prevents the formation of the building blocks. So the building blocks that are used to build the peptidoglycan cell wall are Glucknack-Mornack um, units or residues. And in blocking the synthesis of the Mornack um, unit, the functional building block is not uh, completed. And therefore, we do not see a functional building block being produced with phosphomycin. In this diagram, we see the different drug classes I mentioned in their site of action. We can see that cycloserine and phosphomycin being active in the cytoplasm. We see beta-lactams and bacitracin being active more in the cell wall space outside of the bacterial cell membrane. And in the cell wall space, they are able to inhibit transpeptidase and able to inhibit the shuttle, the C55P, the bactoprenol shuttle. And we see vancomycin being able to inhibit on both sides, inhibiting the D-alanine terminus on both sides, preventing the attachment of further polypeptides and it being active on both the cell wall space, in both the cell wall space and the cytoplasm. Another important drug class mechanism of action is the inhibition of protein translation. Note that protein translation is evolutionarily conserved across species, even between bacteria and humans. What do I mean by this? We as humans, we have eukaryotic RNA. We have ATS, ribosomal RNA, rRNA, ATS. The bacterial or the prokaryotic ribosomal RNA is the 70S subunit. But even though we are eukaryotic, humans are eukaryotic, we still possess prokaryotic or bacterial ribosomal RNA. That is because our mitochondria has its own nucleic acid, has its own genetic material. The mitochondrial ribosomal RNA is similar to the prokaryotic ribosomal RNA, meaning 70S din. The mitochondrial 7 rRNA is also a 70S subunit. The mitochondrial rRNA is composed of a 50S and a 30S subunit. This is different from the eukaryotic ribosomal RNA that we humans generally have in most of our outside in our cells, which is composed of the 60S and 40S subunit. So this is one of those hints that point to a certain evolutionary picture, because archaea, so certain bacteria that are more categorically parang primitive, although they are identified to be more specialized depending on the environment. In any case, archaeal ribosome is more similar to the eukaryotic ribosomal RNA, while the bacterial or the prokaryotic RNA is more similar to the prokaryotic rRNA. So you can say that in a eukaryotic cell, there is some similarity to both archaea and bacteria. In this graphic, we see the different mechanism, different sites of action of some drug classes in relation to the prokaryotic ribosome. We can see here lincosamides, streptogramins, chloramphenicol, and macrolides attacking the 50S subunit, while oxazolidinones bind to the 23S subunit of the 50S subunit, while tetracyclines attack the 30S subunit. But all of these drug classes and all of these drugs, in effect, block protein synthesis, block protein translation by preventing the attachment of your tRNA to your polypeptide, to your growing polypeptide chain. They could do that by occupying the A site or the P site, or they can alter the shape of the subunit and of the subunit, and therefore make it difficult for tRNA to be in the right position. In effect, there is no transpeptidation, there is no protein translation, there is an overall slowdown of the bacterial metabolism. There is a prolongation of the stationary phase. So this is a table listing down the various drug targets and the drug and an example drug class. So we can see here tetracyclines targeting the 30S subunit. We can see here oxazolidinones targeting the 23S subunit of the 50S component. And the most popular in the sense that more drug classes are targeting this subunit, is the 50S subunit of the 70S ribosome. This is targeted by macrolides, chloramphenicol, streptogramins, and lincosamides. And so this is the last slide for this part. So we have identified the cell wall and the bacterial ribosome to be important targets by antimicrobials. But note, in the fight against antimicrobial resistance, there are many other targets that new drugs can exploit to help us control bacterial growth. We can have efflux pumps inhibitors. For example, capsaicin, which you can find in a favorite of mine, the chili, and these efflux pump inhibitors are able to block the bacterial cell from expelling drugs. In this example, we have the NOR-A pump that can expel ciprofloxacin. And therefore, in expelling ciprofloxacin, a quinolone, um, the active effect of the quinolone is neutralized because it's vomited out in effect. It's expelled by the bacterial cell. By inhibiting the efflux pump, the antimicrobial can remain within the cell and therefore be more effective. Other targets or other mechanisms of action to control bacteria would be permeabilizers. And permeabilizers essentially soften or they create small holes in the cell wall, and these allow solutes to move more freely inside from the inside to the outside, and in the process disrupt normal cell function because the solute environment is disrupted, and make um, other antibiotics more effective by weakening the cell wall a bit further. And therefore, in weakening the cell wall, you can see penicillins more effectively bursting the cell open. Another mechanism of action against antimicrobes would be to use quorum sensing inhibitors. As we have partly touched before, partly tackled, certain virulence factors are only expressed when the bacterial cell recognizes it is in a colony or in a biofilm. When the bacterial cell recognizes that, wait, I'm all alone, I'm not surrounded by other bacteria, I'm going to be more inert. Meaning, it's going to wait for a more friendly environment, for a more friendly surrounding, for it to express virulence factors. So, quorum sensing inhibitors, in effect, convinces the bacterial cell that it is alone, that it is not in a biofilm, that it is not surrounded by other bacteria. Therefore, virulence factors are not expressed more actively. And an inert bacterial cell is more likely to be phagocytosed because it is not actively producing toxins. Therefore, it is not recruiting the immune system to be active. Other mechanisms would be in controlling antimicrobial effects would be the use of antibodies that can be specific to bacterial toxins, and the use of type 3 secretion systems. Type 3 secretion system inhibitors essentially prevent a type of a parang a drain or a type of a pump from expelling content similar to the effect of pump inhibitors. And so, um, there are other mechanisms of action for antimicrobials, but for the rest of this lecture, we'll be focusing mainly on two: cell wall disruption and inhibition of protein synthesis. So that's it for the first part. I'll see you on the next part in which we will be discussing cell wall disruption and the drugs that cause it.